Hi everybody and welcome back to this short course of Bioinorganic chemistry. We have been discussing about our life with dioxygen. The great oxidizing power of dioxygen is utilized in respiration to produce huge amount of energy, and cytochrome C oxidase is the terminal member of this respiratory chain, in which this dioxygen converts to water with the help of 4 proton and 4 electron. This is what is the reaction. So, O2 converts to water with a potential of 0.815 volt. This reaction of dioxygen converted to water with the help of 4 proton and 4 electron represents the major source of energy when coupled with the oxidation of electron rich organic food. And as you know that respiration produces huge amount of energy in the form of ATP. I have discussed all this in details in my last lecture. Dioxygen reduction center in cytochrome C oxidase is shown over here once again, and as one see that this heme which is 5 coordinated and copper B which is 3 coordinated can indeed bind dioxygen, and X structure is shown here, and when it binds to dioxygen the X structure also been reported, and one can see that dioxygen binds between iron and copper, and thereby this O bond get cleaved and it eventually converts to water. Unless one give 4 proton and 4 electron all at a time, dioxygen will not convert to water. Indeed, if one do stepwise reduction, then dioxygen produces superoxide, peroxide and OH radical, which are indeed extremely harmful for our life. These are called reactive oxygen species ROS, and which are actually responsible for aging and age related diseases in our body. So, I have discussed all this in my last lecture. Now, superoxide dismutase, which actually convert superoxide into oxygen and peroxides. As you can see that this is a disproportionation reaction or dismutation reaction and thus spontaneous. So, superoxide getting oxidized to dioxygen, and also another molecule of superoxide getting reduced it. And although it is a spontaneous process, nature need to design an enzyme so that the superoxide can be destroyed almost immediately, and as one can see that K catalyst is 2 into 10 to the power 9 mole inverse second inverse, very high values. So, that this has been designed by our mother nature to destroy superoxide almost immediately it produce in our body. The superoxide, as you also can see, produce peroxide, which is another harmful product. This peroxides is an undesired intermediate and extremely harmful for biology, and nature cleverly make use of these peroxides to produce lots of very important chemical transformations, which is otherwise impossible to do. And as you can see that catalase, peroxidase, chloroperoxidase, lignin peroxidase and cytochrome C peroxidases are designed, which eventually convert these peroxides to something very useful products which is otherwise impossible. I also have discussed in details the action of catalase and peroxidase in my last lecture. As one can see that hydrogen peroxide oxidize iron 3 centers to iron 4 oxocation radical, which then converts to iron 3 plus heme centers. In case of catalase enzyme, the hydrogen peroxide is the substrate which is converted to oxygen and 2 H plus. In case of peroxidase enzyme, the organic substrate is getting oxidized to form A. Now, I will talk about oxygenase enzymatic system. The oxygenase enzymes catalyze reactions of dioxin with organic substrates in which oxygen atom forms dioxygen are incorporated into the final oxidized product. There are two types of oxygenase reactions. One is dioxygenase reaction in which both oxygen atoms of dioxygen are incorporated into a single substrate. The other is monooxygenation reaction in which only one oxygen atom of dioxygen is incorporated into the major substrate. So, you see that this labeled oxygen which is red in color in mono and di, they are actually incorporated inside the substrate, and that is what dioxygenase and monooxygenase names come from. Let us talk little bit about dioxygenase. Dioxygenase enzymes are known that contains heme, non-heme iron, copper or manganese. Here is a typical example of risky dioxygenase. As you can see that the benzene converted to catechol in presence of dioxygen, and this catechol. So, this catechol dioxygenase is responsible for cleaving this catechol. It can cleave in two ways. One is extradiol, outside the diol over here, extradiol dioxygenase, which yields a product. You can see that the ring is getting cleaved completely in presence of oxygen, and iron 2 is involved in the enzymatic system, whereas interdiol dioxygenase, it cleaved between two diol group, and iron 3 ion is involved in the enzymatic system, and the products are quite different. So, based on the position of the cleavage, the products are different, whether it is an extradiol or intradiol. Now, I will talk about monooxygenase enzymatic system. For example, cytochrome P450, a typical example of monooxygenase. It is an heme containing enzyme whose active site resembles the dioxygen binding sites of myoglobin or hemoglobin in many aspects, except the axial ligand to iron. Indeed, in cytochrome P450, cysteine sulfur involved, and rather than an imidazole side chain from histidine, which is involved in hemoglobin or myoglobin. Now, the typical reaction is shown over here, where you can see RH substrate converted to ROH, and which is water soluble. And next is tyrosinase, the another monooxygenase enzyme which contains two copper ions in close proximity in its active site, which has deoxy, oxy and mates that closely resemble comparable state of hemocyanin in their spectroscopic properties. And as you can see the typical reactions like that catechol converts to quinone, and typically tyrosinase does this kind of conversion. Next, monooxygenase is methane monooxygenase. There are many monooxygenase known in the literature now. Now, methane monooxygenase contains 2 non-heme iron in close proximity, and it resembles closely to hemerythrin in many of its spectroscopic properties. And the typical reactions that methane monooxygenase does is NADH plus methane plus dioxygen in the help of proton, NAD plus and methanol and water. So, methane converts to methanol, that is why it is called it methane monooxygenase. In addition to these 3, there are also a large number of monooxygenase enzyme known today, which contains non-heme iron or copper ions, sometimes even manganese. So, I am not going to discuss all in details because of time shortage, and today I am going to discuss one of these very popular monooxygenase enzyme, cytochrome P450. Cytochrome P450, as I have discussed already, that RH, the substrate converts to ROH with the help of oxygen, 2 electron and 2 proton, which eventually forms water. And this is a very important enzyme for our body, for our life. It is responsible for the oxidative metabolism of drugs, steroids and carcinogen. It is also a part of the body's detoxification system, hydroxylating compounds, making them water soluble, so that it can easily excrete it through urine. In other words, it protects our body from the attack of foreign chemicals. So, this is very, very important for our day to day life, and the active site structure of the cytochrome P450 in the resting state is like this. As you can see, a porphyrin group is ligated with iron 3 state, and 5th position is cysteine sulfur, and the 6th position is weakly coordinated water molecule. The X structure is also shown over here. Why we call this enzyme as cytochrome P450? This is unusual name is given to it. The reason is that it exhibits an intense Horet band at unusual 450 nanometer for its ferrous carbon monoxide derivative. Typically, for normal heme centers, this generally comes around 420. So, since it is an unusual position, so when it was discovered, it was given a peculiar name cytochrome P450. P is a pigment. So, this is the way it has been named since it was discovered. However, this enzyme does lots of chemical transformations in our body. As I have said already, this is a group of monooxygenase enzyme, and a varieties of substrate being hydroxylated. For example, this camphor is hydroxylated over here, the next steroid is hydroxylated over here, this has been hydroxylated here. So, a typical positions, very selective, not random. Please note that this hydroxylation is stereospecific, not random, and that is the reason why this is so special. The enzyme catalyzes a wide varieties of reaction. For example, some of these reactions are listed over here, like hydroxylation of aliphatic compound CH converted to COH, hydroxylation of aromatic rings, like if you have in benzene, it converts to phenyl. Epoxidation of olefins, the olefin get converted to epoxides as shown. Amine oxidations to amine epoxides N to NO, sulphide oxidations to sulfoxides SO, and oxidative dealkylation of heteroatoms. For example, if it is PHOMe, PHOH plus HCHO. So, like there are many such reactions I can write, which is basically catalyzed by cytochrome P450 in our body. This is what is the protein structure of cytochrome P450. As you can see that at the center there is an heme group, and the fifth position is ligated with cysteine sulfur and sixth position is ligated with a water molecules. What is interesting to note over here is a huge protein chains is wrapping around this molecule. And what these protein chains are doing? The protein chains are actually dictating the reactivity and also the specificity of the substrate hydroxylation, the position of the hydroxylation and other things which I will soon discuss. However, the basic chemical transformation is taking place at the iron center, that hydroxylation, and thus this iron is so important. If we remove this iron, this enzyme is completely inactive. That shows the importance of having iron in cytochrome P450. Now, once I remove this protein chain, what you see that? You see this molecule that heme centers, iron is sitting at the center and there is a weak coordination of a water molecule and the cysteine residue is over here, and this is the molecule which is actually responsible for so many organic transformation. Let us look at that the cytochrome P450 with camphor as a substrate. Now, you see that this molecule is crystallized with camphor which is substrate, and camphor sits just on the top of this iron center and very close to that, and also there is no water molecule which was earlier present. So, that this camphor replaces this water and plays a particular position, so that it can be hydroxylated at a particular position, and this is what is happening here. You see, when I remove this protein chain, this camphor is sitting at the top of this iron center and this carbon is actually hydroxylated, which we will see soon. So, this is the camphor and this position is hydroxylated, and also it is very stereospecific, and that is what the entire protein chains are doing. So, protein chains are responsible for the stereospecificity and also controlling the rate of these transformations. Okay. So, we will see in next few slides more details about it. So, in the catalytic cycle of this enzyme is shown over here. Now, in the resting state, this is what it is in the resting state. This iron 3 center and cysteine sulfur is at the 5th position and 6th position is a weak water molecule, indeed a low spin species, S is equal to 1/2. Now, when a substrate RH comes close to this molecule, then what would happen? It replaces this water and it forms a enzyme substrate complex as it is shown. However, there is a remarkable change in the spin state. The iron center which was low spin in the resting state now becomes high spin, since it is a 5 coordinated iron centers, although oxidation states remains the iron 3. Now, in the next step, this iron 3 undergo a quick reduction in presence of an electron, and this iron 3 transform to iron 2, and this also another high spin complex. Now, once it forms this iron 2, it is ready to bind dioxygen, as we have seen in case of hemoglobin and myoglobin. Indeed, this is what is happening. So, iron 2 binds dioxygen, and as you know that then it will become iron 3 O2 minus, but in presence of one more electron and proton, this becomes O2 2 minus. Okay? It is peroxides. And as I have discussed earlier, that if OO bond need to be cleaved, peroxide is the best state, because the OO bond dissociation energy is lowest. So, immediately the OO bond get cleaved and it forms iron 4 oxo cation radical, which is called compound 1. Please note that this is a very reactive and unstable intermediate, and this also forms with RH. It immediately transform to ROH, and iron 4 is reduced to iron 3. And in the next steps, once it forms ROH, then water molecule replaces that ROH, and this comes back to the original molecule, that iron 3 with an axial ligand of water, and this is low spin complex which is in the resting state, and the catalytic cycle starts once again. During this transformation, what is exactly happening? This RH, the substrate, is getting hydroxylated, converting to ROH in presence of oxygen and proton and electron, and oxygen getting converted to also water along with ROH. So, this is monooxygenase reaction, and this is very important. So, all this molecule getting hydroxylated, and we can use this iron 4 oxo cation radicals for various catalytic applications in the laboratory as well, since this is a highly reactive and very strong oxidant. So, many people, many research group utilize this molecule for transforming lots of organic substrate into various species in the laboratory. So, key step is this iron 4 oxo, and you can see that lots of substrate can be hydroxylated, and this oxo group can be inserted, whether it is an epoxidation, whether it is inoxidation, whether it is aliphatic hydroxylation, whether it is oxidative deamination, whether it is aromatic hydroxylation. Large number of substrate can indeed be converted to various important and interesting molecules, which is otherwise very difficult to make. Okay. So, basically the organic CH substrate converted to COH during these transformations. So, now I will show you a beautiful design principle that is adopted in biology. Now, we already discussed catalase, peroxidase and cytochrome P450 and their catalytic transformations. And now, I will show you that Although the catalytic process are very similar, they are doing completely different job in biology. As you know that catalase which actually catalyze the conversion of hydrogen peroxide to oxygen and water. Peroxidase actually oxidize the substrate, but all these reactions are not oxotransfer reaction, they are basically that the electron transfer reaction. However, cytochrome P450 we have just seen that substrate oxidation is taking place. This is due to the design principle. Although in all three enzymes the common active intermediate is iron 4 oxo cation radical, but they are doing completely different reactions in the biological system. The design is such that in cytochrome P450 this iron 4 oxo is exposed to the substrate, so that substrate can come close to the oxo group which is highly reactive and transform to hydroxylated compound. Now, this protein chains which are actually wrapping around this cytochrome P450 indeed allowing the substrate to come close to oxo group, thereby this oxo transfer is possible. In contrast, in case of catalase and peroxidase, the protein chains which are actually blocking this oxo group, so that substrate cannot come close to the oxo group. However, substrate can come through the porphyrin periphery, it can come close to the porphyrin periphery, and as you can see that the porphyrin is cation radical. So, this radical transfer reactions are actually possible in case of catalase and peroxidase, and that is the reason why these two are doing a completely different reactions, not oxo transfer reaction, whereas cytochrome P450 undergo oxo transfer reaction. So, you see that the beauty of the design in biology here, although they are very similar or they are almost identical in their reactive intermediate, but they are doing completely different jobs in biological system. And here you see that how the protein chains which are wrapping around this molecule are actually controlling the reaction and specificity of the reaction. I have discussed today about the reactions of dioxygen with organic substrates catalyzed by oxygenase enzymes, in which oxygen atoms from dioxygen are incorporated into the final oxidized products that are very useful for our daily life. Dioxin gets activated and then inserted into the substrates, which fall into two categories depending upon whether both or only one of the atoms of dioxin molecule ends up in the substrate. Although a large number of monooxygenase and dioxygenase are known today, we could only able to discuss one such monooxygenase, cytochrome P450 here today due to short duration of this course. In my next lecture, I will discuss various dioxin carrying proteins in biology. Thank you.